Okay, so now, after this amazing lightning talk, I have the pleasure to introduce to you our final keynote speaker for today, Maria Emilia Machado Walter. She's a researcher at the University of Brasilia. She's obviously located in Brazil. She did her undergraduate in mathematics at the University of Brasilia. Then she did a master's in mathematics in the same place. Then she did a PhD in computer sciences in the State University of Campinas. And finally, she did a postdoctoral internship at University Claude Bernard. So now she's a adjunct professor of the University of Brasilia since 1988. And she's the Dean of Research and Innovation in the same university from 2017. So thank you, Maria, for being here today with us. It's our pleasure to have you. We can see your slides now.
So good afternoon. Can you see my presentation?
We see the presentation in PDF, but it's not full screen.
Okay. So, yeah. So, first of all, I would like to thank the organizers to have invited me to this important event. I will talk to you about in-silico methods for analyzing non-coding RNAs. I will talk to you about 4 topics. First, I will introduce what are non-coding RNAs and how we annotate our non-coding RNAs. Next, I will present very quickly in some in silico or computational methods and also databases that are commonly used to annotate non-coding RNAs. I will discuss 2 projects that we have been involved at my university, and after I will conclude. So I apologize to the biologists, but I have to begin with some computational view of, okay, so I don't know exactly, so in eukaryotes, the figure shows this, we have a cell with nucleus, and this nucleus contains DNA that are packed into chromosomes, as you can see. And then the number of chromosomes is characteristic of the species. In humans, we have 46, in fruit flies 8, and in Pica pastoris, that is a fungus, we have 4. Next, please. Next, please. So the chromosomes carry hereditary information, and you can see in the figure, the DNA is stored in a double strand, and this information is used in biological functions in living organisms like growth, development, and reproduction. And so this DNA information produces proteins, and you can see also in the figure on your right, portions of the DNA carry information that is extracted by a molecule of RNA, and this molecule together with other specific molecules are used to produce proteins. Next, please. So we can see this in a computational point of view, and a biological one, of course. We have the protein coding RNAs that are the messenger RNAs, and again, we can see in this figure the double strand of DNA that with a process of the transcription process carry information that are expressed in the cell, and with this proteins can be produced, but some of these molecules of RNA don't code for proteins, and we call it non-protein coding RNAs. Next, please. In this talk, we are concentrated in this task of annotation. To annotate sequences means to find or to derive a specific feature in sequences of DNA, RNA, or proteins. that informs the structure or function of these sequences in the molecular system. The annotation of proteins or finding these molecular functions in proteins is based on methods that work very well and are broadly used. And an example is BLAST. The BLAST tool produces alignment of sequences, but the annotation of non-coding RNA or the finding of specific features in non-coding RNAs don't work so well. And this is the concern that we have in this presentation. We can divide in a general the non-coding RNA in two sets, the small or short non-coding RNAs and the long non-coding RNAs. And annotation can be performed by two processes that are called classification and identification. Next, please. Here we see some classes of small non-coding RNAs. There are many known classes of these molecules, and in this table we see in the first column some classes of these small non-coding RNAs together with the function shown in the second column. So we have, for example, microRNAs that is a family of genes that have regulatory and post-translational functions, and there is SNRNAs or small nuclear RNAs that make modifications of ribosomal RNAs. And there are other classes, but I will talk only about these first two. Next, please. We have some other classes of long non-coding RNAs. They are quite different. There are 6 categories that are known already, and you can see in the figure in green, we see the long non-coding RNA. For example, the first class is the intergenic long non-coding RNA that are a sequence that occurs between 2 coding genes, and these blue and yellow are respectively exons and introns. We have intronic long non-coding RNAs and so on. Next, please. In the classification progress, we want to identify or to classify or to find the classes of small or long non-coding RNAs. And when we want to identify non-coding RNAs, this means that we scan genomes to find these molecules, and genomes here depends on the data you have, so you can have the entire chromosomes or scaffolds or contigs, and this was strongly improved with the RNA-seq, that is a sequencing technology, and it's used to produce transcriptomes or sets of transcripts that are molecules expressed, as I said before. It's important to say that in transcriptome analysis, this identification or the annotation of non-coding RNAs in silico supports laboratory experiments, or the biologists have to prove that this is really true. Next, please. We have some characteristics and challenges in annotating non-coding RNAs. I will discuss 3 of them. The first one is to predict secondary structures from primary structures. You can see in the figure that we have the test sequence is the sequence of nucleotides that form this sequence, and from this we want to infer the secondary structure or a 2-dimensional shape of these molecules. On the left, you have the base pairing and other structures in this small non-coding RNA, and on the right, you have also associated, this is in silico methods, the probability of these shapes are correct. So when you have red, you have a high probability that this is correct. Next, please. The second challenge is to compare secondary structures, and there are algorithms that allow you to do this. In this figure, you see the multiple alignment of six sequences in the top, and you can see this alignment in secondary structures. Next. So we can classify and identify both the sets of small non-coding RNAs and long non-coding RNAs. And here, as examples of these small RNAs, I mentioned this in the table previously, and so we have two molecules here of microRNAs in the left, and the CD box that is and the HACA box that are these two figures on your right. To classify and identify long non-coding RNAs, to do this, we use very different methods. Usually we find them using the length of the sequence. Usually the long non-coding RNAs has more than 200 nucleotides, and some are very high. The length of the sequence is very higher than that, and the fact that they have a very poor capacity of synthesizing proteins. Next, please. Next. So long non-coding RNAs are really difficult to annotate, both in classification and identifications. They are not well known, but today there are many of these identified molecules associated with diseases, especially cancer. Next, please. I will show you some paradigms and tools that we use to annotate non-coding RNAs. The first paradigm is homology. Homology is a biological concept that means descending from a common ancestor. It can be inferred computationally by measuring how similar are the sequences. And this inference is done by comparing genomes of two or more species. This comparison depends on curated databases and usually is done by a query sequence of the organism that you are interested in, and you have a database of sequences with already known functions, and you want to find similar sequences in the databases of the sequence you are interested in. Similar sequences means that they probably have the same biological function as inherited from a common ancestor, and we annotate the sequence transferring the function of the similar sequence to the one that you are investigating. Very well-known examples are BLAST, and that I mentioned it before, it's very known, and Inferno. Inferno means inference of RNA molecules, and this follow essentially the homology paradigm. Next, please. The other paradigm is class prediction that usually is performed with methods of machine learning, mainly with the supervised learning model. Differently from the homology paradigm, it uses ab initio characteristics, which means you extract some characteristic directly from the sequence and don't use this concept of homology. Usually you construct two sets, a positive one with a non-set of non-coding RNAs, and the negative set that it can be formed with proteins, for example, or other sequences, depends on the case of the molecules or the class of the non-coding RNAs you are investigating. And an example that uses this paradigm is CPC2, that is investigate the capacity of coding protein. Next, please. Another paradigm is known as de novo, when you use models distinct from homology and class prediction, a very well packaged that is broadly used to annotate non-coding and to infer secondary structures, and these things that I showed you previously is the Vienna. Vienna is composed of many packages which with methods that are based in thermodynamics, and so they try to include in the computational model biological characteristics like energy. Next, please. We also can you we proposed a different paradigm based on multi-agent systems where we annotate non-coding RNAs as the system be a human annotator. We included many tools and databases and some inference roles that simulates the reasoning, the human reasoning. And this is the method we developed here in collaboration with the University of Leipzig, called ncRNA agents. Next, please. Prediction of long non-coding RNAs are performed in a different way, completely different ways. So we have methods based in support vector machine, like ncRNA scan, SVM, and icRNA. We have methods based on bicolored networks, for example, LNC-GFP, but we have many workflows and pipelines that try to annotate or to find these long non-coding RNAs. Next, please. We also developed a method that is a workflow, but it's based on SVM. We proposed this some years ago. Next, please. And to identify, this is all these tools I told you before, they are tools to classify non-coding RNAs. To identify non-coding, we have some, and I mention here two, that identifies SNORNAs. This is why I mentioned microRNAs and SNORNAs. The first one is SNO-seeker, that identify or scan genomes to track SNORNAs in eukaryotes, and SNO-report that was first proposed by Hertel in Leipzig, and also we at the university collaborated with them to improve this method, and we inserted this SVM also. This was a method to also to identify SNORNAs organisms. Next, please. Following, I will show you some databases that have different classes, and I will show you some. The first one is non-code, that is automatically found. This non-coding were automatically extracted from the literature and gene bank. RNADB that contains the mirror base that contains microRNAs. Next, please. This SNO-RNADB contains SNORNAs of many organisms. Then you can see that you have databases with general non-coding, and some one are specific, like this one, that contains SNORNAs of plants, archaea, fungi, or the plant SNORNA database that contains SNORNAs of plants. Next, please. A very well-known database is RFAM. RFAM is used together with this Inferno method, and it's in its version 14.8, and it contains more than 4,000 families. And with RFAM, you can search and you can make many other investigation. Next, please, including you can extract some information as shown here in this slide. Next, please. For long non-coding RNAs, we have also, as for short ones, we have specific and general databases. This first one is in red, that contains long non-coding of the genomes of human and mouse. The Diana LNC base that provides interactions of miRNAs and long non-coding RNAs. They contain lots of sequences in interactions and things like that. Next, please. The long non-coding RNA disease contains long, as the name says, long non-coding RNAs associated to diseases, and LNC-RNADB contains many sequences of eukaryotic long non-coding RNAs. Next, please. Then I will very briefly show you two projects that we have been studying at the University of Brasilia in collaboration with people in Germany at the University of Leipzig. The first one is this CRC project, where we investigate some aspects of colorectal cancer, CRC. This cancer presents unique characteristics and treatment responses to different sites. You can see in the figure where are these sites that are colon, rectum, and rectosigmoid junction. They are shown in the figure, and you can see a tumor. This is a cancer that affects millions of people around the world, and the treatment is very hard. So erroneous diagnosis of the CRC site can lead to overtreatment with chemotherapy that strongly affects the health of the patient, and specific and common anatomical site biological mechanisms are not well known. So next, please. We tried to investigate some small networks that are called siRNAs, competing endogenous RNA that express interactions of proteins, microRNAs, and long non-coding RNAs. So this figure shows a bit about this interaction. I don't have time to go into details here. Next, please. And so what we did, we proposed a method where we extracted information from a database called TCGA that contains clinical and molecular information about this cancer. We first found some siRNA networks. These are small networks, and some of them were already proposed in the literature, but we proposed news, and with some analysis, we proposed putative prognosis biomarkers. Of course, all of these have to be confirmed in the laboratories. Next, please. Then the next slide shows this this small siRNA in the different sites of this CRC. And the next slide shows the biomarkers that we proposed. This was published into last year. And so we proposed some biomarkers in the different sites. The intention here is to collaborate to understand these mechanisms in CRC, and we are collaborating with a doctor in Brasilia that is work with this cancer in a hospital in Brasilia. The next slide shows, I think, the other project. We wanted to investigate the performance of machine learning methods. They produce good results, but sometimes they are not very good to infer homology in non-coding RNAs. And here the idea is to propose a method to generate arbitrarily large and diverse data sets of artificial non-coding RNAs, and we used this to systematic evaluate these machine learning techniques, most considering large evolutionary distances. Then, so the evolution of these non-coding RNAs were simulated along a randomly in silico generating phylogenetic tree, so we have control of what's going on, and we worked with point mutations, substitution, insertion, and deletion. Next, please.
So the Dr. Walter, we only have a couple of minutes left, so I don't know if you would like to wrap up. Unfortunately, we missed some time due to the outage.
Sorry?
So if you want to wrap up, because we have a couple of minutes left.
Yeah. I'm just finishing. So the goal was to investigate the performance of this machine learning method to predict this coding RNA. You can go to the next, please. We compared this with the BLAST method. BLAST produces very poor results. And the next slides show that machine learning methods, next please. These results showed that these machine learning methods are very competitive and produce good results. You can go directly to the next please. We made some experiments. Next. Then we learned that annotation of SNORNAs using homology inference methods based on primary sequences BLAST leads to poor results, and machine learning methods are very competitive, but it's highly dependent of the how good and how large is the database. So going to the conclusion, can you go a slide? Yeah, and only to finish this, some technical aspects. Non-coding RNAs are important molecules since they play essential roles in cellular mechanisms. Computational methods can efficiently support to predict non-coding RNA function, but these have to be confirmed in wet labs. There are many techniques of distinct areas of computer science, and we have many databases containing different classes of non-coding RNAs. And we can use this because it's publicly available. And the last slide. Some general reflections for these projects. We have a keyword that is collaboration. We have to be involved in multidisciplinary projects that are essential in science, like we faced with the COVID-19 pandemic, since it involves large volumes of data and exact and life sciences. And we have interesting projects like how to train scientists, how to encourage interdisciplinary research in this world of so big and complex problems. And finally, in the context with this event, how to attract women for bioinformatics. And with this, next slide, I would like to thank to my collaborators of the University of Leipzig, Peter, my colleagues at the University of Brasilia, Marcelo and Célia, to Valdeir, and to my students, Daniel, João Vitor, Lucas, and Mirelli. And thank you for your attention.
Thank you. It was an amazing presentation. I think we all enjoyed it. We only have maybe time for one question. So Carolina Canetieri has, and also probably me and many others, a question about what happens when two tools give you different results or contrasting results. When you merge this information into a machine learning procedure, how can different results be merged into something that actually gives you biological insight?
Yeah, this is usual in these projects, and usually you do exactly like this. We investigate different sources, different methods, different databases, and this is the sense and the motivation that we proposed our method. This is exactly this what we did, artificially speaking, in the multi-agent system. And there are some biological roles that are used, and we offer these different solutions to the biologists, and the final word is from them. And so they have to investigate, and my presentation is much for the computer scientists, of course. Biologist that is my friend told me, oh, you have a very particular way of seeing biology. This is why I always apologize to the biologists. But the thing is, we offer this different. Sometimes we can do this in silico. Sometimes we have to have the expertise of biologists to see exactly, and even so, many times they are a bit confused about how what is exactly the function or the biological functions, or I mean how these molecules perform or act in the cell.
Thanks. It was amazing, and it goes again into collaboration. And so I want to thank you for being today with us, all our lightning talk speakers as well. And again, this is about collaboration, and this group is really into fostering collaboration. So please keep connected, keep participating in the Q&A and the chat. And I have one message for the community. So we have on the 23rd of September is the open data days, and the prizes will be announced then in the after the Gender and Science Conversatory. We also have a keynote that day, so even if you see that it's a short day with workshops and the Editatona, do join the keynote about data feminism and the conversatory about gender and science. And then at the end, you get to know if you got a prize for your talk. I want to thank again all the speakers we had today, my co-chair Jalvi, and all the organizers, Lilian, Ana Julia, and Elin Tepa that is also here, and Gabriela that's also helping sharing on their days. So thank you everyone, and I'll let our main organizers close the call or move us to the workshops. I don't know how that works.
Yeah, thank you everyone. We are moving to the workshop. Thank you, Maria, amazing job, and thank you for being here, Alejandra too. And okay, see you tomorrow in the next day of the conference. And for the one who are joining us the workshops, then see you in a while. Bye. Thank you everyone.